So chapter 11 is really about describing patterns of inheritance. So 11.1 is really a lot of background terminology and a little bit on Mendel's experiments. And then we'll talk about patterns of inheritance like complete dominance, incomplete dominance, codominance, pleiotropy, epistasis, polygenic inheritance, and then also how the environment can impact your phenotype, and a little bit about pedigrees and carriers as well. Before we even really knew much about genetics at all, people were still interested in trying to determine how traits were passed from parents to offspring. There was two kind of ideas. The blending hypothesis was the idea that genetic material from the two parents kind of blended together the way like blue and yellow paint would blend to make green paint. Or there was the particulate hypothesis, which is the idea that parents pass on discrete heritable units, which we now know today are genes. And Gregor Mendel documented this mechanism through his experiments with garden peas. So broadly speaking, if we know how different traits are inherited and their patterns of inheritance, we can predict the outcome of a mating between two organisms, or we can at least predict the possible outcomes of the mating between two organisms. So the super brief story on Mendel's experiment is that he discovered the basic principles of heredity by breeding garden peas in carefully planned experiments. He controlled the mating between plants, and he tracked only characters that occurred in two distinct alternative forms. So like white flowers or purple flowers, or round seeds or wrinkled seeds, not like dark purple, light purple, and white or something like that, or short plants or tall plants, not short, medium, and tall plants. He used varieties that were true breeding, so that means that the plants, like purple flowered plants, produce purple flowered plants, white flowered plants would produce white flowered plants, and so on and so forth. So he mated two contrasting true breeding varieties in a process that's called hybridization. Well, I don't expect you to memorize the traits that Mendel looked at, but I just want you to kind of look at them and appreciate it. Like the traits were either tall or short, or inflated or constricted pods. There was no in between there. So he picked good characteristics to look at. And also if we think about the organisms he picked to study, he picked a good organism to study. It's relatively easy to control which pea plants are mating with each other. They have a short generation time. They have a really high reproductive rate, and they're easy to care for. Like imagine if he had picked elephants. Obviously he wouldn't pick elephants, but their generation time is very long. They have few offspring and they eat a ton of food. Even if he picked, more common livestock animals, you'd run into the same kinds of problems. But picking the pea plant was a really good choice. And then picking these particular traits to track was another good choice. So when we talk about genetic crosses, there's different words for the particular generations. So the first generation that we're going to cross is called the P generation. I think of that as like the parent generation. And then the next generation, the parent's offspring, is called the F1 generation. And morals aside, if you take members from the F1 generation and cross them together, so breed brother and sister together, you get the F2 generation. I pick the pictures of dogs because I think, people get the idea of making a Labradoodle, whether you agree or not. I mean, that doesn't really matter at this point, but if you have a lab and a poodle and you breed them together, you get F1 Labradoodles. So they're called F1 because they're the offspring of a lab and a poodle. So what kind of cell division produces gametes? That's kind of a trick question, isn't it? So what kind of cell division produces gametes in animals? Is it mitosis or meiosis? So in animals, meiosis produces gametes, but what about plants? In plants, mitosis produces gametes because, remember the gametified generation in plants from last chapter? That's the haploid generation of plants. And so to go from haploid to haploid, you would do mitosis. So meiosis for animals and mitosis for plants. And why is reducing the number of chromosomes in gametes important? So why can't we have diploid gametes? So if we had diploid gametes and they fuse together, we'd get like foreign, which is tetraploid. So then we'd have tetraploid eggs that would grow up into tetraploid adults. And then when they would breed, foreign plus foreign would be 8N. So I guess octoploid eggs would result, and so on and so forth there. So it's important that we reduce the chromosome number in the gametes in order to maintain the chromosomes in the species from one generation to the next. So then we've talked about alleles a little bit before, but we've not dedicated a whole slide to them. So alleles are alternative states of a gene. So there's a picture of homologous chromosomes, in this case for a plant, and I think we've used this picture before even as well, but it has an allele for purple flowers on one gene or on one chromosome and an allele for white flowers on the other chromosome. So that's like the locus for flower color gene, so where the flower color gene is located, and there's alternative states, either purple or white. So alleles are just different versions of a gene. So in diploid organisms like humans, each individual is going to inherit one allele from each parent. So we have 2 alleles for every trait. So then when we talk about dominant or recessive alleles or traits, when there are 2 different alleles for a gene in the same individual, typically only one allele is going to be expressed or shown in the organism. The dominant allele is always going to be expressed if it's present, and a recessive allele is only expressed if there are no dominant alleles present. So in our flower color example, in this case, the allele for purple flowers is dominant over the allele for white flowers, so we get a plant with purple flowers. In order to get a plant with white flowers, we'd have to have 2 copies of the allele for white flowers. So we use pedigrees and statistical testing to track patterns of inheritance. When we're reading a pedigree, men are depicted by squares and women are circles, and then if we have an individual that has the trait of interest, we'll shade that individual in. And then if you have a square and a circle connected and a line off of them, the line off of them represents their offspring. And when you look at enough pedigrees, it gets to be easy to kind of figure out which pattern of inheritance you're looking at. But until then, you're kind of going to have to guess and check. So what's the difference between a gene and an allele? Really, an allele is just a version of a gene. It's an alternate state of a gene. So like you could have the gene for flower color and the allele for white flowers, or the allele for purple flowers. Number 4, an individual that is heterozygous for a characteristic has what for that trait? Do they have the same 2 alleles or 2 different alleles? So hetero is different, so B, 2 different alleles. And then if an individual is homozygous for a characteristic, that means they have the same 2 alleles for that trait, because again, homo is same. So hetero, different, so 2 different alleles, homo, same, so 2 of the same alleles. So an individual's genotype, is it their genetic makeup or the way their genetic makeup affects their physical makeup? The genotype is their genetic makeup. So like if they have big A, little a, that would be genotype. Or you could say they're homozygous recessive, that would be genotype. Or you could say they're heterozygous, that could be another way to talk about genotype. And then an individual's phenotype is going to be the way their genetic makeup affects their physical makeup. And, you know, genotype is the basis for the phenotype, but the phenotype is not solely the product of the genotype. We'll talk about that a little bit later when we talk about kind of environmental influence on genotype and gene expression. Then what is a carrier? So a carrier is if we have an individual that's heterozygous for like a homozygous recessive condition. So if they're heterozygous, they would be like big A, little a, and then the recessive disorder would have to be like little a. And so if they're a carrier, they carry allele, or they carry the version of the gene, but they don't have that genetic disorder themselves or that trait of interest. So we can use Punnett squares to get mathematical ratios of the possible genotypes of the offspring. This particular Punnett square represents a monohybrid genetic cross. Genetic cross means that we're simulating a fertilization event or a hybridization. And monohybrid, mono is 1, so that means we're only looking at one trait at a time in this case. As far as making a Punnett square goes, you're going to draw a box, and then one parent's alleles are going to go up across the top of the box, and the other parent's alleles will go along the side of the box, and then you're going to drag letters from the top down to the boxes below. And then you're going to drag letters from the side to the boxes beside them. And then you're going to have the possible F1 offspring inside of the Punnett square. So if you had the alleles from the egg on top, the alleles from the sperm on the side here, these are the various potential offspring that could be produced. So when it comes to making Punnett squares, I've got a couple suggestions for you. When we pick which letter to use, realistically, many genes have kind of accepted abbreviations and letters for them, but I don't expect you to memorize them. So instead of memorizing all these different things, let's instead choose the first letter of the word that describes the dominant phenotype to represent the trait, although be careful with your letter choice because you want to have a clear distinction between the uppercase letter and the lowercase letter. So, like, capital P and lowercase p look pretty similar in my terrible handwriting, same with W's and C's, although you can do some things to make that stand out. Like if I was going to use a P, I would write my capital P in print, and then I would do the lowercase p in cursive just to make them look more different. And again, we're going to use the capital letter to represent the dominant allele, and we're going to use the lowercase letter or the little letter to represent the recessive allele. And quite honestly, I know that the capital letter is the big letter and the lowercase little letter is the little letter, but usually, like when I learned genetics and everyone else I've ever heard teach about it says big letter, little letter. So I will use that for the rest of this. So as far as which letter to go first, you should put the uppercase letter or the big letter first. So big B, little b, not little b, big B. When you do problems, it just makes it easier to tell, that big B and little b are the same as little b and big B. So for example, if we were looking at flower color, so that's the flower color gene, and there's 2 alleles, and red is dominant to white, you should choose R instead of W because R is going to code for red, which is the dominant trait. So use big R to represent red and little r to represent white. If you use different letters, it's confusing. So in some cases, there are genes that are named and accepted that use different letters, but at this point in time, use the same letter. So big R for red and little r for white, not big R for red and little w for white. So when Mendel did his experiment with the pea plants, he came up with a law of segregation to describe how gametes pass on traits. So the law of segregation says that each individual has 2 factors, which we now know are alleles for each trait, and the factors are going to segregate or separate during the formation of gametes, and the gamete contains only 1 factor from each pair of factors. So we talked about this last chapter when we talked about meiosis. So we know that fertilization, when the sperm and egg get together, gives each individual 2 new factors. Sorry, not 2 new factors, just 2 factors for each trait. So the law of independent assortment then says that each pair of factors is going to separate or sort independently without regard to how the others separate, and that all possible combinations of factors can occur in the gametes. And we know that happens during which phase of meiosis? So it happens in meiosis 1. Do you remember which phase of meiosis 1? What happens in metaphase of meiosis 1. So this is just showing you the law of segregation, where 2 alleles for each gene are going to separate, and then the law of independent assortment, where the alleles of genes on non-homologous chromosomes are going to assort independently. And they're showing you the P generation, the F1 generation, and the F2 generation, where again, P is the parent or the first generation where we did the cross, the F1 is their offspring, and then the F2 is the offspring of the offspring. So independent assortment, segregation, and inheritance really follow the rules of probability. The outcome of any particular coin toss or any particular fertilization event is unaffected by what happened on previous coin tosses or previous fertilization events. So if we think of the coin toss analogy here, when you flip a coin, you're either going to get heads or tails. So that's like the 2 alleles that the coin can provide. So if we had, sperm, let's say they could have big R for heads or little r for tails, and an egg, we could either have a big R egg or a little r egg. So we have a 1 in 4 chance of getting big R, so having heads right after heads. We have a 1 in 4 chance of getting little r, so having tails right after tails, and we have a 2 in 4 chance of having 1 coin have heads and 1 coin have tails. So it's the same idea with inheritance. It follows the rules of probability. So earlier we talked about complete dominance, where the phenotypes of homozygous dominant individuals and heterozygous individuals are identical, but really there's degrees of dominance. So we're going to talk about complete dominance again, then we're going to talk about incomplete dominance, where the phenotype of the F1 hybrids is kind of somewhere between. It's like a blend between the phenotypes of 2 different homozygous parental varieties. And then we're going to talk about codominance, where we have more than 1 allele that is dominant, and when both alleles are present, they're going to both be fully expressed, and they're going to affect the phenotype in kind of separate and distinguishable ways. So we'll go with examples of each of these coming up. So complete dominance or simple Mendelian inheritance describes a situation in which one gene has 2 alleles and one's dominant and the other is going to be recessive. So the dominant allele will completely mask the presence of a recessive allele. So in our example here, we've got a male plant that's heterozygous and a female plant that's heterozygous. And so the pollen could produce sperm with either big B or little b, and really it's not the pistil, but anyhow, the female gametophyte generation could produce eggs that are either big B or little b. And, you know, when I first grabbed this picture, I thought it was a really good picture, but it's a little bit tricky because they're like coloring the squares all these different colors. So the Big B, if Big B is coding for purple, Big B would give us a purple flower. And then we have a Big B, little b, which is also a purple flower, but that's a heterozygous individual. And then if we have Big B, little b again, that's another heterozygous individual. And then the last square, we have little b, which would give us homozygous recessive, which would be a white flower. When I first looked at this picture, I thought it was a great picture, but they're doing some weird things with the colors in the background on the plants. I guess they're showing homozygous dominant with a gray square, they're showing the heterozygous individuals with the tan square, and then they're showing the homozygous recessive individual with a purple square, even though that's a white flower. So I feel like they got a little creative with the graphic design there. Incomplete dominance, again, is when we have kind of a blending effect. So when we have one gene that has two alleles, which can be expressed as a blend in a heterozygous. So if we have a red snapdragon and a white snapdragon, the red snapdragon is like CR. So the R is standing for red, the C is standing for color, the white is CW, again, C for color, W for white. If we breed those together, the only possible outcome they could have for offspring is offspring who are CW. So CW, red and white blend together and you get pink. So the F1 generation are all going to be pink. And then if we breed the F1 generation together, they could either give their offspring red alleles or white alleles. And so the F2 generation, we have a 1:2 to 1 ratio of red to pink to white offspring there. So ultimately, with incomplete dominance, the phenotypes of heterozygous individuals are different than either homozygous individual. So co-dominance, co means like together, right? We talked about covalent bonds or cooperating where you operate together. So with codominance, you've got a gene with more than two alleles, and two of those alleles are at least dominant, and when they're both there, they're expressed at the same time. So kind of the hallmark example of this, ABO blood type. So we've already talked about blood type a little bit, where we talked about how you have surface markers that talk about or determine what your blood type is. So blood type B has B surface markers, and A has A surface markers, and AB has both A and B surface markers, and then type O blood has neither A nor B surface markers. So a person could have type A blood, type B blood, or type O blood, or even type AB blood. So let's do a problem with codominance and blood type. So create a Punnett square to show the possible offspring from a type O parent and a type AB parent. The phenotypes for blood type could be type A, and the genotype for that would be either big IA, or big IA, little I, or phenotype could be type B with big IB, or big IB, little I, or type AB where we have big IA, big IB, or O, which is little I. So if we have a type O parent, we're going to have little I, and type AB would be big IA, big IB. So we'll set that up. I put the I's at the top, or the little I for type O at the top, and the big I's with A and B on the left hand side. I just picked that, it doesn't really matter. As far as why we use I with blood type, since it's codominant, you need a letter to represent the dominant and recessive traits. And if you didn't know it needed to be I, you could use any letter. Like I've seen students use B before for a blood, and that would be okay. But they chose I because it stands for immunoglobulin. So immunoglobulin A, immunoglobulin B. So it is important to use the I instead of just doing like A for A and B for B and O for O, because you don't know which one would be dominant or recessive just by looking at the problem. So anyhow, that ramble aside, we've got our little I's and our A's and our B's there. So we're going to start pulling letters across. So we're pulling our A's across. So big IA, big IB, big IB, and then we'll pull our little I's down. So little I, and little I. So now we've got our Punnett square with big IA, little I, and big IB, little I, and big IB, little I. So then number 10, is it possible for a type O parent and a type AB parent to produce a type O child? No, not really, because they can only produce big IA, little I, and big IB, little I. So there you go. Then we've got pleiotropy, which is another pattern of inheritance. In pleiotropy, we have an allele that has more than one effect on the phenotype, so one gene can influence many traits. This is not a Mendelian pattern of inheritance either. So pleiotropic effects are kind of hard to predict because a gene that affects one trait often can perform other unknown functions. But if we can trace multiple symptoms back to one allele, then we can say that has pleiotropic inheritance. So Marfan syndrome is an example of this. This produces abnormal connective tissue. And since you've got connective tissue in all of your different organs, if you have a problem making connective tissue, you're going to have a problem in lots of different organs. And this graph below just shows you, or this figure below just shows you the difficulties in the skeleton and the heart and blood vessels and the eyes and the lungs and the skin, because all of those different organs rely on connective tissue. So polygenic inheritance then is really the most common type of inheritance. Poly is many, so this is multiple genes involved in a single trait, or what we perceive as a single trait. So these traits have a lot of variation in them. If you look at the figure there where we look at human height, some people who are very tall, some people who are very short, but most people are somewhere kind of in the middle, or most people are average height. Another kind of example of polygenic inheritance is eye color, although lots of times people will tell you like brown is dominant and blue is recessive, but you guys have looked at people's eyes and seen that there's more than one kind of brown, and there's, different colors of blue as well. So eye color is really a polygenic trait. Another pattern of inheritance is epistasis. This is when one gene can interfere with the expression of another. So in summer squash, fruit color is controlled by two genes, and the first gene has two alleles, and the second gene has two alleles, and the presence of the dominant allele for gene 1 blocks the expression for gene 2. So gene 1, it's either going to be a white fruit or not white or not colorless, and then gene 2 could either be yellow or green. So if we have a squash that's like big W, big Y, little y, what would it appear to look like? So gene 1 has big W's, so that means it's going to be white, and it doesn't matter what gene 2 says because the white is blocking the expression of either yellow or green. So we're going to have white fruit there. If we look at a big W, little y, what do you think? Again, that big W is blocking the second gene's expression, so that's going to be white as well. And if we had little w, big Y, it's not going to be white. So now we get to see what gene 2 says. Gene 2 has big Y. Big Y stands for yellow, so it's going to be yellow. And then if we had a little w, little y, it's little w's, so it's not white. And it has little y, so it's not yellow, it's going to be green. So that's epistasis, where one gene interferes with the expression of another. So let's look at this. These are examples. The first one is from Wikipedia and it cracks me up. We've got the genes for either blonde hair or red hair and the gene for baldness, and I guess the person is completely hairless in this example from Wikipedia. Like, really, if you're bald, you still have eyebrows and whatnot, but not in Wikipedia. Eyebrows are not a thing, I guess. So here, the gene for baldness is interfering with the expression of the hair color gene. So that's going to be epistasis. The Labrador Retriever example is a little bit more complicated. So take a second to see if you can figure that one out. So you can get a black Labrador if they have at least one big B and at least one big E. Chocolate Labs, or the brown ones, right? They have to have little b, and then at least one big E. And then yellow Labs can have whatever in the B area, but they need to have two little e's. So that is another example of epistasis. So we keep talking about, Mendelian patterns of inheritance and, how the genotype determines the phenotype, but really the environment can also influence the expression of a gene. So the environment can influence the phenotype. I think, it's easy to think about nutrition when you think about this, right? If you have not enough nutrients when you're growing, you could have stunted growth, or if you have too many nutrients, you can gain too much weight, that sort of thing. But kind of an interesting experiment is coat color in Himalayan rabbits and Siamese cats. They have dark points, and it's because there's an allele that produces an enzyme that allows pigment production only at temperatures below 30 degrees Celsius. And I don't know, the things people think of doing, right? So they've shaved this Himalayan rabbit and they've strapped an ice pack to it. And then when its fur grows back where that ice pack was, since it was cooler, that enzyme was active and they end up getting a black patch there. So epigenetics is really the above the genome effects. So this is the study of changes in organisms caused by differences in gene expression rather than differences in the genetic code itself. And I don't know if any of you are identical twins or know any identical twins, but when identical twins are born, they're very, very similar. They have, the same DNA. However, as they change, their life experiences are not identical, and so that turns on and off different genes. And maybe you've known of, an identical twin or a pair of identical twins and one's gotten cancer and the other hasn't, or they could just be very different in other ways as well. So I really would encourage you to click on the link when you get a chance and go through the epigenetics in rats experiment. It's basically about how high and low nurtured rats are different in adulthood. So chapter 11 is the first kind of genetics chapter. We're going over how to describe patterns of inheritance. We talked about Mendel's experiments. We talked about a whole bunch of vocabulary and terminology that'll help us understand and set up our problems like homozygous and heterozygous, and the difference between a gene and an allele, and the genotype and phenotype. We talked about how traits or factors are passed on to offspring with the law of segregation and independent assortment, but really those things are happening in meiosis. We talked about dominant and recessive alleles. Be able to do Punnett squares. You'll need to do a Punnett square for a complete dominance problem, or an incomplete dominance problem, or a codominance problem, or even epistasis. Don't worry about doing a Punnett square for pleiotropy and polygenic inheritance or environmental impact, because you kind of really can't. Make sure you know the difference between P, F1, and F2 generations. Understand that the outcomes of each mating follow the rules of probability, and be able to read a pedigree and know what the word carrier means.